Well, it's a pleasure to be here, and I want to thank the organizers for having some flexibility with my talk and timing, because I actually came straight from clinic. And that's part of what I think is a little bit different about my point of view, is I'm a practicing cornea specialist who happens to be trapped in a commercial guy's body, and ultimately provide a view from two different angles here. And so I want to start with a few objectives. Objective number one is I want you to know who Versea Health is. It's a holding company, and this particular company is playing in a variety of different areas, one of which is in drug development and looking at cellular responses to therapies. It's also playing in the world of orthopedics and spine surgery in a way with biologics that it's relatively novel, and bone growth opportunities. It also has a classic point-of-care professional and OTC diagnostics division. And then what I think is my favorite is the Ophthalmics division. And similar to Tarsus, we're building an all-star team to really bring products to market. And from my perspective, it's all about bringing products that actually change clinical management and therapeutic decisions. I repeat, that's another thing I want you to realize, is that we're coming up with complete solutions that guide decision making. And so with that, I like to frame a conversation. I always think it's easier if we're all on the same page. So there are so many people that have dry eye ocular surface disease. This isn't new to everybody, but I want to point out the fact that a lot of people actually have moderate to severe disease. I'd also like to point out the fact that as clinicians, if we are out there treating people, and we treat people with the classic tiers, and then we start with hygiene, and then we move down the list to an immunomodulator, whether it's cyclosporin, any one of the cyclosporins, and lifitegrast, and potentially pulse steroids and plugs, 37% of people still fail. They just don't feel like they have effective therapy. So I believe if you have the right targeted approach from a diagnostic standpoint, we can lower that number. Can't eliminate it because it's a complex multifactorial disease, but we can get better at treating it. And I think I can show you a potential innovative approach from a healing perspective. So let's start with TPOC, which stands for our tier-based testing system. And I'm very careful not to even say it's diagnostic, because in reality, we're providing a quantitative result. That result is diagnostic in itself, but because we guide therapeutic decisions, you can follow that, you can monitor, you can demonstrate to the patient an efficacy in what you're doing. You get buy-in, so it has a much more broader appeal. And one of the things that makes it different is that it only uses 1 microliter of tear. It provides an answer in 6 to 8 minutes, and we're starting initially with lactoferrin and IgE with MMP9 to follow in about 18 months. And so if we look specifically at lactoferrin, people will say, well, this has been around for a long time. Why now? Why are we using it? And it's because it helps us make a distinction between evaporative and aqueous deficient disease. So if you have an aqueous deficient patient, and you can look at a number such as 0.9 micrograms per liter, you have the ability to say, hey, this person would probably benefit from an either immunomodulator or a plug. And you make that determination based on how much MMP9 they have. High MMP9, you probably don't want to put a plug in and pool immuno agents there, like inflammatory proteins. Yet, if they have very low levels of immunoproteins, then they're an ideal candidate for a plug. High levels, you want to go immunomodulator all the way. So it gives you a way to actually think about the patient. Similarly, IgE. What people probably don't realize is about 17 to 18% of dry eye patients are actually allergic, masquerading as dry eye. Or in cases where people have really dry eyes, they don't have the tear clearance to get rid of the allergens so easily. So then they get an exacerbation of the allergic response. So now imagine if you had a threshold, roughly 80 nanograms per ml. You'd be able to say this person has allergic conjunctivitis. We would manage these people differently, maybe a mast cell stabilizer. Or maybe if they had a low lactoferrin and a high IgE, we'd start thinking about a steroid. It's a perfect combination for this particular group of people. Again, we can guide therapy. So then if you flash forward and say, well, what about a red eye? Someone comes in, is it allergic conjunctivitis? Is it viral conjunctivitis? Well, we have thresholds that really tell you whether or not you're dealing with a viral condition or an allergic condition. So it really makes a big difference. Similarly, if we have a way to figure out the right diagnosis or using a testing platform to not only diagnose but to monitor and guide treatment, what better than to have some novel technology that we can use? And so this is always a hard one because there's so much historical information out there and there are a lot of great products, right? I don't think there's a silver bullet in any way, either on the diagnostic side or on the therapeutic, but I think it's situational. And part of being a good clinician is to figure out where things fit and how to optimize your patient results. So if you think about the classic scenario, you have cryopreserved grafts, and these have some great features because they transfer pentraxin and high-chain hyaluronic acid, and they also have some limitations. They have a ring that people don't like, and they also have to be kept cold. At the same time, if you think about it, there's only so much material that can transfer in a micron or in several micron disc. It's going to leach out relatively quickly. And then from a dehydrated perspective, there are advantages from storage and shelf life, but there's potentially some limitations on how the patient feels in terms of comfort. And so here at Versea, we have a very novel approach. We basically said, we don't want to transfer any cells. We don't want to transfer any material, so it's completely dehydrated and decellular. But we took 3 layers of amnion and laminated it together. Why is that important? Because actually, the basement membrane is the scaffold for healing. That is the key. That is where all of the machinery lies. You have laminin, you have fibronectin, and you have all the cells that turn on when you actually get recipient cells to populate it. So next point I want everyone to remember, and that is cell attachment is a natural stimulus for the release of growth factors and cytokines. So we can't do a head-to-head prospective study with another graft, so we do the next best thing, right? You take conjunctival and corneal cells and just corneal cells and epithelial cells, and then you take a classic dehydrated, you take a cryopreserved, and you take a Biovance and you put it onto these media. And you say, how are they affected? And let's look at the data that just published. The very first bar in each of the graphs is actually, represents dehydrated decellular, which would be Biovance. The next is dehydrated, and the third is the cryopreserved. And in the very first slide up there, you're looking at, in purple, corneal and conjunctival cells. In green, these are just cornea cells, and then the last one's just epithelial. So if what I'm saying is true, and you imagine you freeze something, you actually kill all the cells. Cells are dead. You dehydrate something, you kill all the cells. But imagine that you have to have vacancy for where those cells sit to turn on the machinery. So the faster that the recipient cells populate those cells, turn on the machinery, the more organized response you're going to have to healing, because the quick transfer of cytokines and hormones just disappears. And you can see that if you look at the second chart, we have the greatest level of adhesion. And if that's true, you should see a lot of proliferation, which you do. And if you look at day 4, you have the most organized cellular response. And by that, you can see a lot more of the green cells. You don't have the blank open areas. And this suggests that over time, you have this organized healing. And if we look at the cytokines themselves, starting on day 2 through day 7, there's a much more higher quantity of all the critical cytokines. So it's a more organized healing. So when you put it all together, again, you have different needs for different patients. There isn't one specific winner in every circumstance. But we think we have a way to provide a unique 3-layer design that has advantages. The advantages include that most people don't need a second graft. You can do it in one. It's thin, but because it's thin in 3 layers of amnion, you have to be smart about how you apply it. You have to use a bandage contact lens, and you want to pick one that has a high base curve that's flat, so you allow the oxygenation so you don't get any tight lens syndromes. You want it to be on the patient for 5 to 7 days, and then you get a full result. Ultimately, it's easy to handle. Because it's all basement membrane, it doesn't curl up on you. And then we have the ability to basically leave it as room temperature shelf life. It's decellular, so we're not worried about prions or transferring immunogenic material. And it really protects the cornea or conjunctiva. And it's ringless, so it's a little bit more comfortable if you get the right contact lens fitting. It's bidirectional, so you don't have to worry about which side's up, which side's down. And then we get all the classic dehydrated advantages. So I kind of think of it as somewhat of a hybrid. So taken together, we really have at Versea a combination of diagnostics that drive clinical management and therapeutic decisions. We know we can improve the quality of patient management, and for those patients that actually don't improve enough, we believe we have a novel amniotic membrane approach that can really lead to better overall care. Thank you, and both Rahim, Healthegy, and I will stay after in the back if anyone would like to chat.